Mutation is a change in DNA sequence. It can result from radiation, exposure to chemical agents, or infection by viruses. When it occurs in the sex cells, it can be passed on to offspring, while somatic mutation occur in body cells are not passed on. They can cause cancers and congenital malformations. We have three most important gene mutation types: point mutations, frameshift mutations, and trinucleotide repeat mutations. Point mutations are a change in a single nucleotide of DNA. It can have one of three effects. First, the base substitution can be a silent mutation, where the altered codon corresponds to the same amino acid. In missense mutation, the altered codon matches with a different amino acid. On third, the base substitution can be a nonsense mutation, where the altered codon corresponds to a stop signal. Stop codons are UAA, UAG, and UGA. In this picture on the bottom, you can see nonsense mutation, where the altered codon corresponds to a stop codon UAA, so the protein synthesis will stop. A frameshift mutation is a particular type of mutation involving the insertion or deletion of a nucleotide in which the number of deleted base pairs is not divisible by 3. That's important because the cell reads a gene in groups of 3 bases. Every group of 3 bases corresponds to one different amino acid that are used to make proteins. If a mutation disrupts one of those reading frames so that the wrong amino acid is put in place, then the entire DNA sequence following the mutation will be disrupted or read incorrectly. Trinucleotide repeat mutations or dynamic mutations occur when the number of triplets present in a mutated gene is greater than the number found in normal genes. They share nucleotides G and C in their sequence. One of the examples is Fragile X syndrome, which has 200 to 4,000 repeats of CGG within a gene called FMR1. The number of triplets continues to increase as a disease gene is inherited, and that's why we have normal persons, carriers, and affected persons within a generation. Look at this picture. You can see a reverse triangle, which the tip is toward normal range of trinucleotide and the base reflects the mutation range, which is increased during gametogenesis. As you can see in this slide, CAG trinucleotide is repeats, adds a string of glutamine to the proteins that is happen in Huntington disease. These are more examples of diseases caused from trinucleotide repeat mutations.